Hello everyone, my name is Autumn Rogers and I'm a sophomore immunology and medical microbiology student, and I did my project on the analysis of circulating microRNAs targeting DNA methyltransferases as a predictor for treatment outcome. So in our project, we looked at two different epigenetic molecules. The first one is microRNA. It starts out on the nucleus as pre-microRNA, and then these two proteins, DG-CRB and DROSHA, allow it to be cut and processed. And then exportin 5 exports it out of the nucleus into the cytoplasm. And then once it's in the cytoplasm, dicer comes along and it allows it to become mature microRNA. Then the 3 to 5 prime strand of the mature microRNA gets targeted by this RISC complex, which is comprised of the RISC protein and another protein. This is important for the microRNA because it allows it to go to its target gene. Once it's at its target gene, either transcription stops or ribosomal activity stops. So either way, no functional protein is being made. The next epigenetic molecule are DNMTs, or DNA methyltransferases. There are three functional types of DNMTs. DNMT1 is a maintenance molecule, and DNMT3A and 3B are de novo methyltransferases. As you can see in this figure, there is a huge difference between the normal cell DNA methylation and a cancer cell DNA methylation. These bubble-like things right here are called CpG islands, and they're in the promoter region. As you can see in the normal cell, there is hypomethylation, or not a lot of DNA methylation in the region. And in the cancer cell, there's hypermethylation, or a lot of methylation in the region. So how does methylation occur? A protein recruits DNMT, and DNMT comes in and methylates the cytosine of the CpG island. When there's methylation on the CpG island, RNA polymerase cannot bind, and transcription cannot occur. And in cancer cells, a lot of the times we see that tumor suppressor genes cannot be transcribed, which leads to cancer. So how are these two related? MicroRNAs are known to maybe target DNMTs, so that's why we're looking at them in our project. How do we know which microRNAs to analyze? We used a website called microRNA.org, and it allowed us to see which microRNAs, or miRs, had the best binding affinity for the different DNMTs. So we saw that miR148A has good binding affinity for DNMT1, and miR29A has good binding affinity for DNMT3A and 3B. We also did miR16 as a positive control because it is known to be elevated in the plasma of cancer patients. So we started out with whole blood patient samples, and we used a blood plasma collection tube. Basically, this separates the whole blood from the plasma, as you can see on the bottom and the top. And then we took the plasma. After we had the plasma, we isolated the RNA using the RNA mini kit from QIAGEN. Basically, cells were lysed and the RNA was bound to the membrane. And then the membrane was washed with the wash buffer and ethanol. The RNA was eluted with deionized water, and then we used this machine right here called the nanodrop. It's basically a fancy spectrophotometer connected to a computer, and it not only allowed us to see the concentration of our RNA, but the purity. Once we had pure isolated RNA, we made cDNA, or complementary DNA. Basically, a PCR reaction was set up using a master mix comprised of primers, nucleotides, buffers, enzymes, and the microRNA primers, and then we used reverse transcription to make the microRNA into the complementary DNA. Once we had cDNA, we diluted it and did real-time PCR. Basically, this is just another PCR reaction using the specific primers, miR16, miR148A, miR29A, in addition to nucleotides, buffers, and cybergreen, which allowed us to see fluorescence, and enzymes. Once we had data from the real-time PCR, we calculated the average CT value, or the copies per reaction, for the sample, and then we calculated delta CT, which is the difference between the samples and our internal control. And then we used delta CT to calculate the relative copy number by using the formula 2 to the negative delta CT. So in this figure, you can see our results. On the y-axis, you see the relative copy number, and on the x-axis, you see our patient samples, which is 8 in total. Right here, you can see that miR16 was highly expressed as we expected because it was our positive control, whereas miR29A and miR148A were downregulated compared to miR16. So in conclusion, miR16 was highly expressed in patient plasma, which matched the literature. The expression of miR148A and miR29A was downregulated as compared to miR16. And this data suggests that miR29A allowed DNMT3A and 3B to be highly expressed in plasma, and miR148A allowed DNMT1 to be elevated in plasma or the cellular component of patient samples. So all of this data is taken pre-treatment, so pre-radiation, pre-chemotherapy. So we want to look at the data after treatment and compare it with treatment outcome. We also want to compare this data with a healthy donor. And we are currently working on increasing our sample size. I just want to thank the UBank lab for allowing me to work on this project with them.